Hello, my name is Keith Choate, and I will be reviewing the slides that I presented at the Foundation for Ichthyosis and Related Skin Types listening session. I'm the Associate Dean for Physician Scientist Development, a professor of dermatology, pathology, and genetics, and the Director of Research in Dermatology at the Yale University School of Medicine. So to begin, I'll tell you a bit about the National Registry for Ichthyosis and Related Skin Types. The National Registry is a partnership between my research group at Yale and the Foundation. It includes a total number of registrants of about 1,300, and remarkably, we've been able to provide genetic diagnoses to about 1,030 of these families. All samples that we receive are pre-screened for mutations in 51 genes. And for those individuals for whom we don't find known mutations, we undergo whole exome sequencing or whole genome sequencing. And our goal is ultimately to get a genetic answer for every single individual who's enrolled within the registry. One of the wonderful byproducts of having created this registry is we've not only created a great genetic resource, but we've also asked people to provide significant amounts of clinical information. And so what we've done is we've created a genotyped phenotyped resource, which has really enabled a number of clinical and translational studies in disorders of keratinization or cornification. And so if we think about what's contained within the registry itself, it includes subject demographics, it includes medical history data. It includes measures of quality of life and of specific variables, so quality of life indices, including the Dermatology Life Quality Index, the Children's Dermatology Life Quality Index, the Family Dermatology Life Quality Index, as well as 5D itch data. It also has gene-level mutation data. We permit survey integration, that is, surveys can be administered by the registry and can go out to defined populations that go right down to the specific gene of mutation. And all individuals who've enrolled within the registry have been asked if they will permit us to recontact them to update their demographics and also to consent to future studies. So in terms of the registry outcomes, we were able to create two of the most important scoring systems for understanding the severity of ichthyosis, the VIIS in 2019, and the ISS, which was just recently accepted into JAMA Dermatology. We've also contributed to a number of publications, including one which touches on mental health in adults and children, another which touches on the severity of alopecia with ichthyosis. And of course, we've also been able to identify over a dozen new genetic causes of ichthyosis, which has significantly increased our understanding of the fundamental mechanisms that underlie features of this group of disorders. In terms of drug development, we've been able to identify individuals who have rare genotypes to participate in multiple independent clinical trials. We've established cell lines for preclinical development. And of course, we've also developed new therapeutic discoveries, and these include things like cholesterol lovastatin for the treatment of disseminated superficial actinic porokeratosis and ILVEN, and also retinoids for KDSR mutations, which we know induce ceramide biosynthesis and improve the disease state. So we also recognize that there are future challenges in ichthyosis therapeutics. And this includes the fact that we really need to make sure that we're using measures that are actually both important to patients and also matched to genotype. And so things that we think about that are relevant to individuals with ichthyosis include the redness of their skin, the scaliness of their skin, the amount of itch that they experience, pain that they experience, absence of sweating, loss of hair, thickening of the palms and soles, fragility of the skin, skin odor, depression, and quality of life. One measure which is currently assessed in many clinical trials is fissures. The issue is with the definition of fissures. So fissures that are present on the palms and soles are very relevant to individuals who have ichthyosis, but fissures of the skin itself is a measure that's much more difficult to assess. And would not be considered a cardinal issue across the many different genotypes represented within ichthyosis. I think that the next thing that we'd really like the FDA to consider is the fact that localized improvement may vastly change patient experience for a visible disease. That is, if you have redness and scaling that's present on your face, on your arms, and on your palms and soles, having the capacity to just treat those areas would include a measurable improvement in symptoms and potentially in quality of life. And so the FDA is quite used to thinking about 75% improvements of overall disease severity, but in ichthyosis, I think we also need to be thinking carefully about localized disease severity. We also need to think about the timing of the interventions that we make. And so we recognize that there are unique periods of risk depending upon the genetic mutation that underlies an individual disease type. So examples include the perinatal period for harlequin ichthyosis, in which infants are at high risk of infection and mortality. Also, the failure to thrive in childhood for many different subtypes of autosomal recessive congenital ichthyosis. So clearly, there are moments in which action are necessary. And so as the FDA is considering therapeutics, we need to be thinking about moving quickly from adult indications to childhood indications to even indications in the very first moments of life. We also, of course, need to consider the impact of disease upon children. We recognize that childhood is a formative period for the development of identity. And if safety is clear, as I mentioned before, it's absolutely critical that we move trials to children early with the hope that we might be able to improve their quality of life and to help them through important developmental stages. So going back to what is a meaningful endpoint. So unlike atopic dermatitis and psoriasis, which often affect the entire body, a 2-point improvement in a global assessment may not be possible. And I know that that's the measure that's used for many clinical trials in other disease states. So in talking with my own patients who have participated in clinical trials, I want to communicate to you that they've reported that even modest improvements can have a significant impact on their daily life. And this was with respect to total amounts of redness, time spent grooming on a daily basis in order to feel like they're ready to engage with the world. And so we need to be thinking about outcomes that are meaningful to the patients that we are seeking to serve. And then, of course, I think that it's important for the FDA to recognize that some of the cardinal therapeutics in ichthyosis lack indication. And of course, lacking indication is a central flaw, because as you know, the FDA controls approval of medications, but Medicare will not allow off-label use of medications and certainly won't pay for off-label use of medications. And so despite the fact that retinoids like isotretinoin and acitretin were extensively trialed in ichthyosis, the companies that initially did those trials never sought FDA approval. And the lack of FDA approval has created a dramatic barrier to access for patients, particularly for Medicare. And many of my patients who have Medicare are forced to self-purchase these medications. And so the question that I pose to the FDA is how we might be able to obtain access to these therapeutics, which remain the standard of care for many of our patients within the organization. I'd like to thank you for your attention and for hosting us here from the Foundation for Ichthyosis and Related Skin Types. Your understanding of this disease state is really central to your role in thinking critically about new trials that will come across your desk and about coming up with next generation cures for this important group of disorders.